Let's take a look at the initiation process of RNA transcription. Now, we'll remember that RNA transcription has three main steps. The first of these being initiation, which we'll look at in this video. The second step is elongation, and the third step is termination. Now, the second and third steps, we will talk about them in another video, so stay tuned for that. We've got two diagrams here, and we can see that there's a lot going on, and there's a lot of components that are involved in the initiation step of RNA transcription. So let's talk about those. We've got our DNA molecule here. So we've got our two strands, our template strand and our coding strand. Remember, it's the template strand which is involved in RNA transcription. And we've got this purple part of the DNA molecule up to here, which we'll say is the gene within DNA that we're concerned about in transcription. Now, up from this, we've got an area that's represented as blue, and this is something called the promoter sequence. That's the promoter sequence. And this has a very important role within RNA transcription because it tells RNA polymerase, which is the enzyme involved in the production of mRNA, exactly where to bind on at the top of the gene. Now, within the promoter, we've got this area represented as black, which has a specific relevance, and this is called the Tata box. That's the Tata box. And we'll see exactly what this does in the second diagram, but we want to mention a few other things here. We've got some transcription factors which all help the process of RNA transcription to take place. So we'll just mention a few specifically. There's transcription factor 2D, there's transcription factor 2B, and here we have transcription factor 2H. And we've got a series of general transcription factors is what we'll call them up here. So looking at the bottom diagram, let's talk about what's going on. The transcription factor 2D is the first molecule that's involved. And over here, we'll see that it's got this area that's a different color. And this is called the Tata binding protein, or TBP for short. And as the name suggests, this area is the part of the transcription factor which binds onto the Tata box. And what it does is it causes a local distortion in the shape of the area of DNA at the Tata box. So it essentially acts as a signal for RNA polymerase, which is this molecule here, and that's our enzyme, remember. And it tells RNA polymerase exactly where to join on at the top of the gene. Now, what happens after transcription factor 2D joins onto the Tata box? Well, transcription factor 2B joins on, and RNA polymerase makes its way over to the top of the gene, and we've got our general transcription factors here at the top. The general transcription factors, which are obviously joined on to the RNA polymerase itself. And lastly, we have transcription factor 2H, which we talked about here. Now, this has an important role, and it has two main things that it does. One thing is that the tail of RNA polymerase here, it phosphorylates it, which means it adds on phosphate molecules to it, which essentially activates RNA polymerase, allowing it to start the process of RNA transcription. And what it also does is it separates the two strands of DNA, which exposes the template strand, which is what we're concerned about in RNA transcription.